In this video, we will introduce the computational tools and software associated with TopoTunnel and demonstrate their usage. First, let's take a look at the source code and data. The source code is primarily divided into two main components, the pipeline and the UI. The pipeline handles the tunnel calculation and topological representation. The UI, on the other hand, is responsible for visualizing and analyzing these results. By downloading the source code, we can run the program locally. The Protein MD datasets for TopoTunnel are available for download on Zenodo. In this video, we will use the MD data of the CYP3A4 protein as an example to demonstrate how to use the program. First, let's download and extract these two files. Then, we will run the pipeline program. In the interface, we need to enter the paths for the Protein MD file and the input directory, as well as the name of the output folder. If you already have the Caver tunnel calculation results or a pre-generated reference conformation, you can provide the details for the tunnel profile and reference conformation. Otherwise, the program will generate them automatically. The remaining parameters can be set based on a relationship between the protein in your current MD data and the reference conformation PDB. If we already have the Caver calculation results, we only need to select the data preparation option. However, to demonstrate the entire process from tunnel calculation to mapping calculation, we will select both options. Under the Caver tunnel calculation settings, when the number of task splits is set to 1, the program uses a default calculation method provided by Caver. When the task split number is greater than 1, we provide an accelerated approach based on a divide and conquer strategy for Caver. This method speeds up the tunnel calculation by splitting a single MD trajectory into multiple segments. Next, we need to enter the paths for the downloaded Caver config file and the Caver executable, and then run the program. Once the execution is complete, we will obtain the topologically represented pathway results. The entire program takes about 43 minutes to run. Specifically, the Caver tunnel calculation takes 42 minutes, while the topological representation of the pathways takes about 1 minute. Opening our results folder, we can see that both the Caver calculation results and the topological representation results have been successfully generated. In this section, we will show you how to visualize the calculation results. First, let's launch the UI program and load the folder containing our calculated results. In the center view, we can see the current mapped pathway results. By holding Shift and left-clicking, we can select a specific pathway. The frames in which these pathways appear will then be displayed sequentially on the right panel. By clicking Residues Combination Compare, we can explore the differences in pathway residue pairs across different datasets. Since only one dataset is currently loaded, this self-comparison naturally shows no variations. Furthermore, we can select specific residues to observe the variations in their associated residue pairs. By clicking Residues Compare, we can observe the variations of individual residues. Clicking the Sync Charts button allows us to observe the trends in the radius and other properties of the currently selected pathway. In the Dataset panel, we also provide options for color coding and displaying labels based on data clusters. Next, we import a randomly transformed pathway to observe whether the topological representation is affected by rigid body transformations. As we can see, the two topological representations are completely identical. In contrast, the spatial pathways exhibit significant differences due to the rigid body transformation.